All right, let's go ahead and get started. Few quick announcements. We have the final 3-minute thesis due tonight, so make sure you get that in before the deadline tonight. The next announcement is next Thursday, the 10th, we have our first draft of the research statement due. So that's going to be basically taking a lot of the pieces of what you've been working on and putting it together into a 2-page research statement. You'll be including your figure illustration. You'll be incorporating the method that you submitted in your previous method assignment, for which you'll be getting feedback on by tomorrow. And we'll be providing a couple of examples of what this looks like. But again, it's going to be 2 pages. And the issue should not be how can I extend this into 2 pages, but rather the real challenge is going to be how can I squeeze this into and fit within 2 pages, because that's typically going to be the challenge here. And again, this is going to be fitting the format of the Harrison Undergraduate Research Award as well as the NSF Graduate Research Fellowship. And again, the NSF Graduate Research Fellowship is the most prestigious graduate fellowship award out there. And this could be an opportunity for you to practice writing that for when you're applying to graduate school your 4th year. And if you're fortunate enough to get one, that's basically you having the opportunity to punch your ticket to any graduate program in the country. And so this is an opportunity for you to, again, practice the opportunity to hone the ability of communicating your science and your ideas in the written format. We've been practicing communicating our ideas through a figure illustration, through an oral presentation, and now we're going to be pivoting after the 3-minute thesis to a written format. This is going to be a first draft, not looking for it to be perfect, but rather try and focus on getting all your ideas on paper, try and have a polished first draft, for which, again, we're going to have an opportunity to provide feedback from one another in the format of peer review and submitting a final research statement at the end of the month. Are there any questions on the research statement that will be due next Thursday? There's no questions now. I suspect there will probably be questions in the next week and a half. Okay, and we have our exam on Thursday. And so with that, there's a review session scheduled for tomorrow evening at 6 p.m. Do you happen to know where it's going to be located? Okay. I'll have the members of the teaching team who are organizing the review session tomorrow night to post where it's going to be. But again, we'll have a review session tomorrow evening at 6 p.m. With the location for it to be posted in an announcement tonight. With that being said, we'll go ahead and jump into our content for today. So we've gone over DNA replication, we've gone over transcription, and how transcription is regulated. And so at this point, we have a 5' capped polyadenylated mRNA transcript. And now we're going to be discussing translation and how that mRNA transcript is going to be used to produce a polypeptide, a protein. And we'll be talking about this within the context of ricin. Ricin comes from castor beans, and that bean comes from the castor plant, Ricinus communis. And the castor plant can be found all throughout the southern United States. The plant's depicted here, and it has this colorful fruit on it that possesses the seeds, these castor beans. From the beans, you can extract castor oil, and for some of you that might sound familiar, castor oil has been used for a long time as a laxative. And so it's also used in a few different petrochemicals as well as plastics in various industries. In addition, there's also maybe what you guys have heard of castor bean poisoning. The oil itself is not toxic. While it can be nauseating and can induce and can be used as a laxative, it's not toxic in and of itself, but rather it is when you've harvested the bean and taken out the oil from via an oil extraction, what's left over is this protein-rich water-soluble fraction that possesses a cytotoxic protein. That cytotoxic protein is ricin. There's a fair amount of it. 5% of the dry weight of the castor bean is ricin itself. There's a series of symptoms that are associated with ricin poisoning. And a lot of those symptoms culminate in death within a few days after exposure to even the smallest amounts of the ricin protein. Ricin protein has been used in a variety. The ricin protein has been used has been weaponized in a few different ways. The most high-profile example was the assassination of this Bulgarian dissident by the KGB, Georgy Markov, back in 1978. There was a very small amount added to the tip of an umbrella that was poked into his leg that left him dead several days later after that exposure. And his assassination is kind of what's been used, has been represented in a variety of movies and films and TV shows. And so this was an example of ricin being used and weaponized for an assassination plot. Additionally, it was also used as an attempted assassination where it was sent via mail to President Trump during his first term to the White House, as well as to Secretary of Defense General McMaster, I think it was, as well. And so there's been high-profile attempts of it being used successfully, unfortunately, as well as high-profile attempts of it being used in attempted assassination attempts as well. And so again, the ricin toxin itself, what we'll be focusing on, works by trying to permanently disable ribosomes and therefore being able to permanently and irreversibly disable translation. And so that's the main reason why in which we're going to be using ricin as our application point for translation today. Before we start jumping into the nuts and bolts of translation, I'll stop for any questions here. Yes. No, the effects of ricin on our ribosomes is irreversible, and so there is currently no known treatment for ricin exposure or ricin poisoning. All right, we can go ahead and jump into our content for today. So this might look familiar from our very first lecture back in January, which probably seems eons ago now. But this is what we talked before about how there are a combination of amino acids that are encoded for by these three nucleotides that we refer to as codons. And so and so now that we've been able to go over not just amino acids, but now having gotten to a point where we have a fully mature mRNA transcript, what we can appreciate now is that within an mRNA transcript, that there are three RNA nucleotides that typically encode for and correspond to a specific amino acid. What you might be able to also see is that there are 64 possible combinations. And so we have one of four nucleotides that can possibly be within within an RNA. We have U, C, A, and G, and then three nucleotides within a given codon that encode for a amino acid. And so that equates to 64 possible combinations. And again, this grouping of three RNA nucleotides within the mRNA transcript, we typically refer to as a codon. So a codon will encode for a specific amino acid. And you might also appreciate that there are multiple possible codons that can encode for an amino acid. And there's specific features of this combination that we want to try and take a moment to describe. And so an amino acid can be there's a degenerate quality of the genetic code, meaning that an amino acid can be encoded for by more than one codon. So if you just look at, for example, leucine in the first column, second row, we have, let me get my mouse here. We have four possible combinations, all with a variety in that third spot, where these three different codons can encode for leucine. So again, there's a degenerate quality where an amino acid can be encoded for by multiple different codons. However, it is unambiguous as well, meaning that each codon will always encode for that specific amino acid. So while leucine can be encoded for by CUU, CUC, CUA, or CUG, it's unambiguous, meaning that CUG will always encode for leucine. It will not encode for some different amino acid. Um, the non-overlapping feature of our genetic code also what that means is that when the mRNA transcript is undergoing translation, there's a reading frame that is looking at or trying to read three base pairs at a time. That's why we have codons. There's three nucleotides within this mRNA transcript that is establishing what our reading frame is, and why three at a time will make more sense once we start looking at ribosomes. But again, when we talk about this reading frame, it just means that how many nucleotides in this mRNA transcript are being looked at, and it's three bases at a time, and we're shifting over the reading frame three at a time. And so while 61 codons encode for specific amino acids here, we have also three codons that do not encode for an amino acid, but rather are what we call stop codons. They will encode for the termination of translation and lead to the termination of synthesizing this polypeptide during translation. While it's April Fool's Day and I could make this terrible joke of you have to memorize all of this, I wouldn't do that to you because I've done that in previous years and they didn't find that very funny. What I will say is that there is one codon that you do need to memorize and do need to be mindful of, and it's the one circled here in blue. It's AUG. That is our start codon, and it encodes for methionine. And that's how we typically have methionine as the first amino acid in any protein, because it is the start codon that is the first it is the first nucleotides established in the mRNA transcript during transcription, and therefore also encodes for, or sorry, forgive me, it is the code for translation to begin at that site, AUG, and encodes for methionine. So that's why every polypeptide starts with methionine. And the easy way to memorize it, AUG, school always starts in August, AUG for August. So that's the one codon that I'll ask you guys to know the codon and know what it encodes for, and know that it's a start codon. So I'm going to kind of go over the big picture of translation, and then we're going to jump into the details of it with a little bit more information. And we're also going to look at it in prokaryotic versus the eukaryotic system. So typically in translation, we have this search that tries to identify where the start codon is. And again, the start codon is AUG. And we'll have the assembly of our large and small ribosomal subunits that come together during initiation to fully come together and form the full ribosome with the small, large subunits together. And the first start codon, we'll have the first transfer RNA bringing in the first amino acid here, which encodes for methionine. And what you can appreciate here is that there's these three sites within the ribosome, and that's what establishes our reading frame. These sites that are within our ribosome are large enough to look at three base pairs or three nucleotides at a time along the mRNA transcript. So what ends up happening is that this is initiation, where we have the identification of where the start codon is, the joining together of the large and small subunits of the ribosomes. And during elongation, what ends up happening is that there's going to be this shifting over with the help of specific proteins that move the ribosome three nucleotides or a codon down the mRNA transcript at a time. And transfer RNAs are coming in, bringing in an amino acid, adding it to the growing polypeptide during elongation, and ejecting out an empty transfer RNA where its amino acid that it shuttles in has been added already to the growing polypeptide. Uh, once we get to one of the stop codons that we saw on the previous slide, once we reach a stop codon, a transfer RNA is no longer coming in, but rather there's a specific protein that helps that recognizes the stop codon and will terminate the process of translation here and promote the release of our complete polypeptide. And so again, this is kind of the steps of initiation, longation, and termination are kind of like our main signal posts to kind of keep in mind of where we are during the process. But we're going to take a little bit of a closer look at each one of these steps to provide a little bit more detail. So first, to kind of take a look at one of the most important actors here during translation is transfer RNA. Each transfer RNA there's going to be a different transfer RNA that that equates to a specific codon, and that'll be bringing in a specific amino acid that corresponds to the codon. We have this acceptor stem here, or sorry, forgive me. We have an anticodon here that is complementary and anti-parallel to the codon that it's reading. So the anticodon right here will be kind of almost base pairing and be anti-parallel and complementary to the codon that's within that reading frame in our ribosome during translation. In addition to that, there is this acceptor stem at the top where the amino acid that it's shuttling in is going to be attached. Excuse me. And there's going to be an ester bond that joins the amino acid at the acceptor site to this transfer RNA. And so there's going to be specific transfer RNAs with an anticodon that corresponds and is anti-parallel to specific codons that bring in the amino acid that corresponds to it based thinking back to that genetic code that we saw a couple slides ago. And so there's going to be different species of transfer RNAs with specific anticodons here. And so this is another depiction of what this transfer RNA looks like, again, with its anticodon down below and our amino acid attachment site here, this acceptor stem at the 3' end up top here. And again, it's going to be, we have this moving, if we look at the transfer RNA starting up here at the top left, we go from the 5' end to the 3' end, and it's going to be anti-parallel to the mRNA. So that means the mRNA is going to be 3' to 5' down below here. And so this transfer RNA, forgive me, encodes for alanine based on the anticodon and what codon it would correspond to. If you're looking down below, you might have seen a few of the different nucleotides that are there, and you might have seen an I there that might have seemed a bit out of place, but there's a specific nucleotide that's unique to transfer RNA that we're going to talk about in a moment. While we have 61 different codons that encode for amino acids, we don't have 61 different transfer RNAs. The reason why is because the third position, or I should say, forgive me, it's the first position in the anticodon and transfer RNA, we call that the wobble base. What that means is that the base pairing there is not strict. And what that means is that there can be a little bit more flexibility between the first codon on the anticodon and the third codon, or sorry, I keep saying codon, forgive me, I'm going to take a step back. What this not strict base pairing means is that the first position on the anticodon is going to bind not very strictly or tightly to the third nucleotide on the codon. So when you think back to when I pointed out on leucine, I'll go back a few slides to the genetic code. So if we look at the first column, second row, we have 4 different possible combinations of what can encode for leucine, and the variability was at the third nucleotide of that codon. That third nucleotide, again, it's going to be anti-parallel and complementary, is going to be binding to the first nucleotide on the anticodon. And so it's going to be not, so this binding is not going to be strict and provide a little bit more flexibility. And what this means is that there's going to be multiple codons that can be recognized by a given transfer RNA. So when we look at anticodons and the base pairing that happens between the anticodon and our codon, what ends up happening is that we'll have base pairing that can happen between from A to U and C to G, but uracil will be able to bind to either A or G. G will be bound to either C or U, and we have this special nucleotide that's on our anticodon that's called inosine that can bind to either U or C or A. So there's a lot of flexibility, particularly when inosine is the first nucleotide in the anticodon. And so if we take a step back, you can see here below that the anticodon is IGC. So this first position is the wobble position, the wobble base, and it's an inosine here, which means it can bind to 3 different possible nucleotides on the codon of our mRNA transcript. And so inosine is only found in transfer RNA. You're not going to find it in DNA. You're not going to find it in mRNA. And it's typically going to be, on average, typically going to be located in that wobble position, that first position on the anticodon, across from the 3rd nucleotide on our codon. And again, it can bind to either U or C or A. And the way inosine is the way it emerges, the way it arises is by a deamination event on adenine that produces the inosine here. And so as an example for the amino acid isoleucine, we have 3 codons that encode for isoleucine. And when we look at the anticodon on the transfer RNA that brings in the isoleucine, the anticodon will encode for IAU. And again, this is anti-parallel, so that means that there's going to be the 3rd nucleotide here is going to correspond to the 1st nucleotide on the codon. The second is going to correspond to the 2nd, and the 1st on the anticodon is going to correspond to the 3rd. And so that's why this inosine here in the wobble position again is the 1st nucleotide in our anticodon, is going to lead to U or C or A being present in the 3rd position on our codon. I'll stop here for any questions that you guys might have. Yes. Yeah, so I'm going to scoop myself here a little bit. But if you think back on the last couple of lectures, or I'll say a couple, I'll maybe go back 2 lectures ago. There are instances in which there can be DNA damage that can occur, where there might be a change, like a nucleotide change in the DNA, that might change what mRNA emerges in the transcript. Even if there's that one nucleotide change, because of this wobble and this flexibility, what ends up happening is that this redundancy allows us to possibly still encode for the same amino acid, even though there might have been a change in the mRNA there. Does that make sense? Yeah. Yes. This slide? Okay.
They'll stay in the zone or with every other
Yeah, we're going to go into greater detail on that. There is basically these 3 compartments within the ribosome. The middle compartment is where the growing polypeptide will be housed, basically. A loaded transfer RNA with amino acid will come into the right compartment. We call it the A site for active, meaning it has an amino acid. The polypeptide will be passed over to that new amino acid that comes in to keep adding, almost like beads on a string. And then the whole ribosome will shift over. And now we'll have an empty, whatever was originally bound here in the middle is now going to be empty. And is going to be kicked out once it's on this far left compartment. Does that help clarify?
Yeah, so what's the right? signal, does it track the tRNA to?
The fact that if that site is empty, there's going to be transfer RNA that are going to be coming in and seeing if they fit and correspond to that codon there.
So just like a bunch of them trying to see if they fit, it's not like a signal.
Yep. Exactly. And there's a couple of proteins that help facilitate this process as well. I'm gonna go here, then there, and then we'll move on. Go ahead.
tRNA differentiate between stop codon and the like cysteine or tryptophan, because I'm looking at the table here and
So the first part of your question is quite easy to answer. We don't have transfer RNAs that correspond to the stop codon. There's going to be a separate there's going to be a totally separate protein that identifies the stop codon and comes in if there's a stop codon at that site. Does that help clarify? You can go ahead.
Regards to waffling, what causes the base pairing for the first one third like not straight?
What caused in that wobble position, what caused it to not be strict? To my knowledge, I think it's a feature of transfer RNA. This is the best answer I have. I think it's a quality or feature of the transfer RNA where within the anticodon, that first position, the nucleotide that's there is, I think, structurally, I don't know if it's because of the hairpin loop, and I actually should have said something about the hairpin loop as well. We have 3 hairpin loops on transfer RNA where there's self-complementarity, just kind of like the hairpin loop with the termination signal in the mRNA transcript. But what I suspect is that the hairpin structure with the anticodon first position creates a little less stability between the anticodon first position and the codon. But that's the best answer I have. I'm not 100% certain. All right, we'll go ahead and press on. So there's a family of enzymes that are tasked with linking together amino acids to the transfer RNAs. Aminoacyl tRNA transferase. Again, the long names of enzymes kind of help describe what the function of that enzyme is. And so they help to load transfer RNA with an amino acid, or another way of describing it is charging the transfer RNA with an amino acid. We typically have 1 transferase for each amino acid. What that means is that there's going to be a specific species of transferase that corresponds to an amino acid and loading it to a given transfer RNA. And so this process of attaching the amino acid to tRNA requires 1 ATP to complete the ester bond, and the energetics of that bond is going to be used to form the peptide bond between adjacent amino acids during translation. And so that we're gonna be able to leverage the energetics of this 1 ATP that creates the ester bond between transfer RNA and amino acid to then use it to create the peptide bond between adjacent amino acids in our growing polypeptide. And so to try and help try and look at and define some regions of the mRNA before translation begins, it's going to be important to do so, kind of comparing the prokaryotic versus eukaryotic mRNA here. We have to the left or upstream of our start codon, we have this region that doesn't really get touched during translation. And so this region to the left of the start codon, we typically refer to as the 5' UTR, a 5' untranslated region. And that's everything before the start codon to the left, towards the 5' end. We typically refer to what's between the start codon and the stop codon as our coding sequence. And that's everything that is going to be contributing to providing the code for what protein is going to be involved or read during translation. And everything to the right of the start codon, or everything downstream from the, sorry, not start codon, everything to the right or downstream from the stop codon we typically refer to as the 3' UTR or the 3' untranslated region. Excuse me. And so you'll typically hear people refer to the 3' UTR or the 5' UTR, and there are specific functions that we've identified over time as to what they do beyond not contributing to translation, but they provide some pretty key functions. And so the 5' UTR typically helps contribute to the kinetics of translation and help provide a regulatory feedback to translation. The 3' UTR is also pretty critical for post-transcriptional modifications. And so while they don't provide direct coding sequences that are read during translation, they provide key regulatory feedback. And so the main structure that's going to be really critical for us during translation is called the ribosome. It's made up of two subunits, the large and the small subunit. And each subunit is comprised of ribosomal protein and ribosomal RNA. And so this is where that third species of RNA comes in, where ribosomal RNA makes up or is a part of the ribosomes that are critical for translation. There's a few key distinctions here between prokaryotic and eukaryotic ribosomes. The bacterial ribosomes are described as the large subunit being 50 Svedberg units and the small being 30 Svedberg units. This isn't necessarily tied to their molecular weight, similarly the way we would talk about kilodaltons in previous lectures, like back during protein biochemistry, but rather Svedberg units typically correspond to how they precipitate out or how they sediment when spun down during extraction. And so, excuse me, the eukaryotic subunits are 60S and 40S, but it's not a clear when you have the fully combined large and small subunits together, that it doesn't equal 80S, again, because it's not about molecular weight, it's about how it pulls down and sediments out during spinning. And so the whole subunit together is 70S, I believe. And eukaryotic, it's 60S and 40S for eukaryotic small and large subunits. And when you have the full subunit together, that spins down to 80S or 80 Svedberg units. There are 4 important sites for us to be mindful of within our ribosomes. The first being the mRNA binding site. That's where, as the name implies, it's where the mRNA binds to our ribosomes. And as I was describing in response to a question earlier, there are these 3 compartments within the fully formed ribosome that are really key. Moving from right to left, we have the A site or the active site or the amino acid site, where we have transfer RNA loaded with an amino acid, a charged transfer RNA, enter into that far right A site, the active site. The middle site is called the P site. It can be referred to as the peptide site or the peptidyl site. That's where typically the growing polypeptide is attached to the transfer RNA within that site. Okay. And the far left site, we typically call the E site. Typically call it the exit site because that's where an empty transfer RNA is going to be present without an amino acid and where it will exit out and leave from the ribosome. And so what ends up happening is that you'll have a loaded charged amino acid transfer RNA come in on the A site. It'll basically have its amino acid tethered and it added to the growing polypeptide. Everything will shift over and it'll be in the P site with that growing peptide. What was in the middle site before is empty now and is in the E site and will be ejected, for lack of a better phrase, from the ribosome during translation. And so it's a continual iterative process where things are constantly being added in, tethered, and kicked out from the ribosome. Did you have a question? Just to make sure I heard your question properly. What prevents another ribosome from binding to the mRNA?
Yeah. While it's
That does happen. And we're going to talk about that in about 8 slides from now, but that does happen, yes. So the initiation process. So this is step 1. we have a family of proteins that help mediate the docking of the small ribosomal subunit onto our mRNA. These are called initiation factors. The nice thing with the proteins that help contribute to these steps here is that their name kind of matches with that step, for lack of a better phrase. And so here, this is going to be first looking at the prokaryotic system, and then we'll shift and look at the eukaryotic system. Within the prokaryotic system, they're just called initiation factors, and they help, again, the small subunit bind to some ribosomal binding site. These ribosomal binding sites are consensus sequences. Again, these are conserved sequences that have been identified bioinformatically. And in this case, they have a name called the Shine-Delgarno sequences. And so what ends up happening is that the small subunit will identify these Shine-Delgarno sequences, these consensus sequences that are a cue for a binding site for this small subunit, small ribosomal subunit. And what ends up happening is that the initiation factors help with that binding. There's going to be GTP that's hydrolyzed and mediating this binding of the small subunit to the ribosomal binding sites. What ends up happening is that once the small subunit is bound and at that first start codon, formulated methionine binds to that start codon. And so going back to our innate immunity lecture, prokaryotes or bacteria, their first codon is typically a formulated methionine. And so that's how they're different from us, where ours is typically methionine. In this case, that first start codon, AUG is going to encode for a formulated methionine, and that's going to bind to the start codon. And there's going to be more GTP that's going to be hydrolyzed to bring in, by initiation factors, to bring in the large subunit to now complete initiation and begin the elongation process of adding in more amino acids to this initiated translation process here. Again, this is within prokaryotes. Within eukaryotes, it's a little different. Again, we do not have a formulated methionine as our first amino acid. It's just a methionine. Our initiation factors have a different name. They're eukaryotic initiation factors. We don't have these consensus sequences upstream from our start codon. We don't have those, eukaryotes don't have the Shine-Delgarno sequences, but rather what ends up happening for initiation in eukaryotes is that there's this forward search. So what ends up happening is that we have a complex of our eukaryotic initiation factors and our small subunit, small ribosomal subunit, forgive me, that starts up at the 5' cap and searches downstream to identify and find the start codon. Once that start codon is found, then the large subunit will come in and bind via another GTP hydrolysis event. So again, just to kind of go back and reiterate this, we don't have these consensus sequences in the eukaryotic system, but rather there's this forward search all the way from the 5' cap with our eukaryotic initiation factors that bring in the small subunit. There's another GTP that's hydrolyzed. Once we get to the start codon, transfer RNA comes in and binds to that start codon. The large subunit is brought in by other initiation factors that, again, hydrolyze another GTP, and that's what completes initiation in the eukaryotic system. I'll give space for any questions here on initiation, these factors, or these ribosomal subunits. Yeah. It comes after the start codon is found. Oh, forgive me. So, sorry. So there is a complex with the transfer RNA that encodes for the thionine. There's the eukaryotic initiation factor transfer RNA that will start searching from the 5' cap. We'll have the small subunit come in that binds as well. Once that start codon is found, then the large subunit comes in. So forgive me, that first transfer RNA is a part of that complex. Yes.
The 5' cap versus 3' for the
Yes. All right, so we can jump into elongation from here. So now that we have our first amino acid, that transfer RNA is going to be present within the P site. And so what ends up happening now is that we have a few other proteins that help contribute to this process. We have the hydrolyzation of more GTPs, and there's going to be proteins that help shift over the ribosome to look at the next codon. So what ends up happening here is that we have the transfer RNA with methionine within the P site. We have these elongation factor proteins. The first one's EFTU that brings in the next transfer RNA to the A site. We'll have two GTPs that are hydrolyzed. The energetics from the ester bond between the transfer RNA and the amino acid is leveraged to then create this peptide bond here between these adjacent amino acids. And that's mediated by peptidyltransferase here. And the way this works is that the amino acid, whether it's 1 or 30, are going to be added on to the amino acid that's brought in at the A site. We have another elongation factor protein that will then basically think of it as like almost like a gear shift that will then rotate and push the ribosome down to the next codon. And what was in the P site is now in the E site. What was in the A site is now in the P site. And so what's in the E site is kicked out. And now we have our growing peptide with attached to a transfer RNA in the P site, an open A site, ready for the next transfer RNA to be brought in. And so to kind of like look at this a little bit closer, we have EFTU, again, it's going to have two GTPs bound to it that is serving as a shuttle for transfer RNAs to bring it in to the A site. It's going to and if it's the right transfer RNA, what ends up happening is that and forgive me, this is what ends up happening is that these two GTPs will end up being hydrolyzed. And this is a depiction of EFTU in the right here, relative to the A site, the P site, and the E site within this fully formed ribosome. And so again, we're going to have these GTPs hydrolyzed, peptide bond formed between these adjacent amino acids. It's going to be passed over and loaded onto the transfer RNA with that new amino acid. And so this is what it'll look like now. EFG is the next elongation factor that will then basically push the ribosomal complex down one codon and shift what was in the A to the P and the P to the E. And so just to kind of help try and describe this via an animation, we have our peptide with that's attached to a transfer RNA within the P site, the middle compartment. EFT will bring in the next transfer RNA amino acid, so it's a charged transfer RNA. If it's the right transfer RNA, it'll end up releasing and docking in. That peptide bond will be formed between those amino acids. EFG will come in and basically shift the entire ribosomal complex down one codon. Now we have in the E site an empty transfer RNA that'll be kicked out. And now we're back to an empty A site, an empty E site, and a transfer RNA with our peptide in the P site. This has also been written out for the sake of studying, but I'm also not going to read all through this for you. But what I will do is I'll give space for a few questions here on either initiation or elongation. Yes. Yeah. Yeah. So we have two GTPs here that are attached to EFTU. If the anticodon that corresponds to this transfer RNA, if it's correct, what ends up happening is that the two GTPs are hydrolyzed here. to enable the transfer RNA to dock and be within the ribosome. Now, the energetics from the ATP to form the ester bond between this amino acid and this transfer RNA, the energetics from that bond will be used to help create the peptide bond between these adjacent amino acids. And then there's another GTP that's hydrolyzed by EFG to move the ribosome down to the next codon, where everything shifts, and now we have an empty transfer RNA within the E site that's ejected out. Sorry? Correct. Correct. Yeah, the energetics for attaching is EFTU attaching a new transfer RNA at the A site. Yeah. Yes.
Recognized in the P site, then after that, all the following neurons are recognized.
Correct. Yeah. All right, we can go ahead and jump to termination. And again, the next slide here is largely for the sake of your studying that kind of helps write it all out. We have the animation for visual learners as well as this slide for those who appreciate reading it in a written form. So the step of termination. Termination occurs when we get to the stop codon showing up within the A site. There was a question earlier about transfer RNAs and how do they recognize the stop codon. We do not have transfer RNAs that recognize the stop codon. What ends up happening is that there's going to be a class of protein called release factors that are tasked with recognizing the stop codon. And so what ends up happening is that the release factor will come in and dock and bind when we have a stop codon within that A site. And there's going to be a water molecule attached to a release factor. What ends up happening there with that water molecule is that when the peptide chain transfers from the P site over to release factor that's in the A site now, that's going to help, that water molecule is going to help create the carboxyl terminus of this peptide chain. And once that happens, that's going to basically lead to the release of the peptide chain. And once that peptide chain is released, the entire complex falls apart and we reach the end of translation. So the key factor here that leads to the end of translation is release factor. It's what recognizes the stop codon once it shows up within the A site. And there's a water molecule attached to it that helps mediate the formation of the carboxyl terminus and leads to the release of this growing polypeptide. Here's an illustration of what the release factor looks like from a structural biology illustration. And the characterization of release factor, I think, won the Nobel about 30 to 40 years ago. And the characterization of the ribosomes and the ribosomal subunits won the Nobel about 30 to 40 years ago as well. And so each of these main concepts that we talk about in class were all awarded Nobels on the order of as recently as 2018 or as far back as 1975. So some odds and ends, some clarifications that kind of touch on some questions that we had throughout the lecture so far. As the polypeptide is being synthesized during translation, the hydrophobic forces that dictate folding in proteins that we talked about in the very first lecture, that is happening as the protein is being synthesized. And so there's going to be folding and structural changes as the protein here is being synthesized during translation. We have one ATP and three GTPs that are used for each amino acid. So we have the ATP for the loading of the amino acid on the transfer RNA. We have 2 GTPs from EFTU and 1 GTP from EFG. And so the energetics involved with adding 1 amino acid to a polypeptide is going to be 1 ATP and 3 GTPs. And this touches on a question a moment ago. What can happen is that we can have many ribosomes binding to along a single mRNA. And so we typically refer to this as polyribosomes, and this maximizes efficiency. And this is particularly the case in the prokaryotic system, where we have an operon, where we have polycistronic mRNA, multiple proteins encoded for on a single mRNA transcript. So in the case of the lac operon, we had beta-galactosidase and galactoside permease. And so we're going to have multiple ribosomes binding at once that are translating simultaneously along that single mRNA transcript. And so that's depicted here, where we have, excuse me, moving from the right end to the left end, many ribosomes binding across it. And you can see these like wiggly lines coming out. That is the polypeptide chain being formed and synthesized off of these ribosomes. Particularly for polycistronic mRNA, again, that's not the case in the eukaryotic system. They will typically have multiple ribosomal binding sites. And so, again, there can be the translation for multiple proteins along one single mRNA that where we have multiple proteins encoded for within polycistronic mRNA. Again, polycistronic meaning more than one protein encoded for, whereas the eukaryotic system, we have monocistronic mRNA typically. There are some exceptions that have been identified, particularly in the case of viruses, but in some cases with eukaryotic mRNA, where there are internal ribosomal entry sites or IRESs that allow and enable translation to occur in a cap-independent manner. But this is primarily the case with viruses. There are very few instances of that happening in the eukaryotic system, but particularly the case within the viral system. So what can end up happening is that, and this touches on a question from earlier, that there can be DNA mutations that change the DNA nucleotides that can then lead to changes down at the mRNA level that lead to changes at the amino acid level. And so there are different categories for these mutations and how these changes occur and how we identify them. So one substitution or mutation that happens is called a missense, where we have a nucleotide change that leads to a change in the amino acid that it encodes for. So we originally had CTT at the DNA level that was mutated to CAT, or a variation to CAT. That changed the mRNA transcript that it encoded for, and that changed the codon and the subsequent amino acid, where we went from glutamate to valine. There are also nonsense mutations where a base change leads to the formation of a stop codon, where we go from AAT to ATT, and that change leads to encoding for this UAA stop codon that'll lead to the abrupt stop of translation occurring for this protein. And so this is if you hear nonsense mutation, you might see that in the literature for some of the papers you're reading. That typically means that there was an early stop codon that led to a shorter or truncated version of that protein. One example of that is the NOD2 lecture back in Innate Immunity, where we talked about how the NOD2 had this insertion deletion that led to a nonsense mutation that then again led to this early stop codon that led to a smaller NOD2 receptor. Finally, the last one is a silent mutation, where we had a base change that led to no changes in the amino acid sequence. And so this is one of the advantages of the degenerate feature of our genetic code, where there might be a mutation that can happen, but that led to no change in the amino acid that it encoded for, and basically protected and maintained the functional amino acids that made up that protein. And so there's also insertions or deletions that can happen. So the previous slide was looking at different substitutions. There can also be insertions or deletions. This can happen. One example of this is from non-homologous end joining, where there's the random sampling of nucleotides that are inserted that lead to perhaps an insertion of an extra nucleotide or the removal of one accidentally that lead to a shift in the reading frame, where we had the original DNA sequence to the left here. And after TAC, there was guanine inserted at the DNA level that led to a change in the codons at the DNA level. So before it would have been TAC, TTC, AAA, CTG. Now it's TAC, GTT, CAA, ACT, and that will lead to a change in the amino acid sequence that then changes to the mRNA sequence that leads to changes at the amino acids that they encode for. This is called a frame shift mutation that oftentimes leads to a nonsense mutation. Again, nonsense is an early stop codon. So there's it led to two missense mutations and an early stop codon of this nonsense mutation. So the key thing to note here is that when there are insertions or deletions, it leads to a frame shift change that oftentimes leads to definite nonsense or definite missense and can lead to an early stop codon with this nonsense mutation. And so it can be helpful to think through and ask yourself, like, what would be the consequences of each of these different mutations? What can be considered in terms of gain of function or loss of function? Loss of function meaning that this protein loses the ability to serve its main function, whereas gain of function typically means that there's a loss in a regulatory feedback on this protein, or it functioning in some new way where it didn't be able to function before. And so thinking through, would a frame shift mutation lead to gain of function or loss of function? Could nonsense or a silent or a missense mutation lead to these different gain of function or loss of functions? Excuse me. And so another way to describe the gain of function is thinking through or being able to think about there's potential loss of the allosteric regulation of that protein that leads to, again, unregulated activity, particularly if this protein that it encodes for is an enzyme. Before we pivot back to ricin, I'll give space for a couple of questions here. Yes. Right. The first change or the primary change happens at the DNA level, and that DNA level change will typically lead to an mRNA change. But the changes we're talking about don't happen primarily at the mRNA level first. Does that answer the question?
Yeah.
There are, let's see. There are some post-transcriptional modifications that can happen. But those modifications don't typically lead to like changes in the nucleotide sequence. It's usually changes in how there's a lecture we're going to have after the exam that basically is like an elaboration on all of this. One of them is kind of like not changing the nucleotides in the RNA and in the mRNA, but rather regulating and changing. What's a good way to say this without jumping into it too far? So typically we get transcription to translation. There's a feedback loop that can stop translation from happening and lead to degradation of the mRNA. But it's not leading to changes in the mRNA sequence. So there are post-transcriptional modifications that can happen that provide a feedback loop, but those modifications are not like actual changes in the mRNA nucleotides, but rather feedback and regulation. But again, any mutations that we talk about, there's a whole we're trying to avoid saying mutation. the variations that happen in the at the DNA level, like we have a whole naming system for that called single nucleotide polymorphism. So polymorphism, rather than mutation, is typically what we say nowadays. So any polymorphism or single nucleotide polymorphism is at the DNA level always. It's reflected at the mRNA level, but happens at the DNA level. Yes.
There's a.
Mhm.
Yeah.
No, so there's typically, so like this is like like a, I don't know if I'd say application, but this is an example like the consequence of like non-homologous end joining, where there's like the random sampling of nucleotides that are put in. And so that's from a double-stranded break. So there's going to be nucleotides added on both strands that create a that are complementary to each other, but not going to create like totally offset strands across the chromosome. If that makes sense. Mhm. What do you mean by that? Well, that would be like the in the slide before that, that would be the like mutation where there's like a single nucleotide change. Oh, I get, you're talking about like the hydrogen bonding, like you're talking about the base pairing when there is that mutation. Um, it's actually a good question. With all of the diseases that are described by like a single nucleotide change, I don't know if I've heard anyone talk. I don't know if I've heard anyone talk about like the change in actual like DNA structure. Um, I suspect there would be like a an alteration in the DNA there physically, but um, I can't say for sure. Um, that you know, we talked about that during replication because there's like an open end in the DNA there, but typically, yeah, typically there is with these. I'll make this simple. With diseases that are described by single nucleotide changes, it's not a it's something that's like genetically encoded that doesn't happen from some mutation event, but rather a mutation event that happens during development. Does that help clarify? So like there won't be like off base pairing, but rather there'll be correct base pairing. But what's supposed to be at that protein, it's expected to be a certain DNA sequence, but it's different. Does that make sense? Like it's like within a given exon, it's supposed to be like CTTA, but in what happens in a person with a specific disease where they have like a single nucleotide change, um, it'll end up being CGGTA or yeah, or CGTA. If that makes sense. Yes. Yeah. These are good questions. Any more questions? All right, we can take it back to ricin now. So ricin is a heterodimeric protein. It means that there's two subunits, ricin A and ricin B. Ricin A is the enzyme and is and the cytotoxin, but it cannot quite get into cells efficiently in and of itself. It needs ricin B. Ricin B is a lectin. Lectins are sugar binding proteins. You can think back to mannose binding lectin during the immunology lecture where we talked about how that family of protein binds specifically to mannose, that family of sugars, mannose binding lectin. In this case, it's ricin B is not cytotoxic, but rather does enter cells efficiently. So ricin B is binding to sugars on the surface of cells and enabling the whole heterodimeric protein to enter the cell. It's also an agglutinin, which means it's a protein that promotes coagulation or aggregation of molecules together. And so both ricin A and ricin B are bound together by a disulfide bond. That'll be important once we talk about how ricin B moves throughout the cell. So the way ricin A and ricin B enters the cell is going to be kind of the opposite path of what we talk about in a few lectures, protein trafficking. Whenever extracellular matrix proteins, cytokines, growth factors, these proteins that are released by cells, they are trafficked out by exocytosis. And so ricin A, the ricin heterodimeric protein, kind of follows the reverse pathway of coming into the cell. So again, ricin B is a sugar binding protein. On the surface of our cells, we have all of these sugar-rich proteins, these glycoproteins, and the ricin B lectin will bind to one of these sugar-modified proteins on the surface of our cell. It'll then get endocytosed and internalized. And what ends up happening is that it's internalized into a vesicle that we refer to as the endosome here. And so this endosome is going to be shuttled through different organelles. Some of it's going to be returned back to the surface and exocytosed. Is going to be exocytosed, but a lot of it will continue to traverse through the cell. Some of it will get degraded in a lysosome, but again, it's going to continue trying to process getting to what's called the Golgi. And so when we're in either this extracellular space or or within the endosome or a vesicle, these are oxidizing environments that will maintain that disulfide bond. Okay. But as some of it continues to either get degraded in a lysosome or some get sent back to the surface, some of it will escape from the Golgi. That's going to be an organelle that we're going to talk a little bit more about when we get to protein trafficking. And it's a critical organelle in shuttling proteins that have just been translated out into the extracellular space. But as it leaves the Golgi, some of it it'll get into the cytoplasm. Once ricin gets into the cytoplasm, we're now in a reducing environment where it'll now undergo and change this disulfide bond, reduce that bond so that ricin A and ricin B will separate now, and ricin A can carry out its cytotoxic function. And again, what that's, I kind of scooped it myself here, but it's oxidizing versus reducing environment. Ricin A, what it does is that it's an enzyme, and what it does is that it specifically depurinates adenines on ribosomal RNA and makes modifications to the ribosomal RNA. And again, that's a key piece of what makes up our ribosomes. And so that modification, this depurination of adenine, will inhibit the large ribosomal subunit from binding to the small subunit, large subunits binding to the elongation factors. That'll eventually lead to the loss of function of our ribosomes. And so because of that, translation will stop completely. That ribosome is irreversibly destroyed. And so with each enzyme, with each molecule of ricin, one ricin A molecule can depurinate up to 50,000 ribosomal RNA molecules. And so, excuse me, and we have about 10 million ribosomes within a given cell. And so with you can do the math of how many, approximately of how many molecules of ricin it would take to kill all of the ribosomal subunits within a cell. But it doesn't take very much. Very little amount of ricin can destroy and halt translation completely within a given cell, thus killing that cell, because it won't be able to create the proteins that are necessary to sustain that cell's life. There are some unique applications of how ricin can be used beyond being weaponized. It can be used in research to study the effects of the mechanisms of translation. There are some labs that have actually carefully used ricin as a tool to study translation, how it works, how it's regulated. It can be used to study how protein binding occurs to nucleic acids, because again, this is one of those instances where we have a protein, a ribosome that binds to a nucleic acid strain, mRNA, and so it can be used to help study that as well. You can envision a way in which this can be used strategically to target cancer cells. If this is such an effective way to stop translation and functionally kill a cell, you could conceivably tether or combine a ricin molecule to a antibody that targets a marker specific to a cancer cell. A lot of cancer cells have unique markers that are found on their surface. Kim Kelly in our department has actually kind of launched her career on discovering a unique glycoprotein that's only on cancer cells, that's not found on normal healthy cells. And so you could conceivably tether a ricin molecule. Sorry, were you saying something?
Nothing more.
Sorry? That's right. Cell surface bucket. Yeah. Sorry, forgive me. Never mind. But yeah, so you can combine it and target a unique marker that's found on the surface of cells. So if translation really caught your attention, we have Dr. Beyer here in the microbiology, immunology, and cancer biology department who studies it. With that, it's 3:15, so I'll go ahead and stop there, but I'll hang back for any questions. Thank you, guys.